So we are the original BioAg. You'll hear of companies called BioAg, but like Gary Zimmer's company is called Midwest BioAg. Dr. Robert Faust founded the company over 40 years ago. So he was able to get a hold of that name, and so that's who we are. And we're a 40-year-old company. Little bit more about me. So finished at Oregon State University with an ag science degree in 1990, and then I went to the family farm. And I was there for over 30 years. In 2020, I retired from the farm. As many of you know, small business, particularly farms, can get a little interesting when it comes to family business. I chose to leave in 2020. Played retired for a year and then started to work with BioAg. It's a little more story around me being introduced to BioAg and Vic Sakhalkar, the owner, CEO of BioAg. In 2017, I had met Dr. Robert Faust. We were leasing the property there at the manufacturing site. We were farming, I don't know, a couple hundred acres in that area near Buena Vista. And I met Dr. Faust. Was my first introduction to carbon farming, humus farming. He wanted to do a little seed treatment, a little winter wheat trial. It was actually rye, fall planted rye. And we stood on the platform of our 1590 no-till drill, and he started talking about humus farming versus chemical farming, right? And all I'd ever known was what the university told us and the agronomists. And he was speaking a language I didn't understand, right? That stayed in my head. And when I knew I was going to work again somewhere, he wasn't the first guy I called, but I think he was the second guy I called. And the rest is history. And my mind has been filled up. My heart is filled up with agriculture again in a whole new way. You know, when you turn 50, you better do something different because your brain's going to just kind of fade away. Greg and Cindy have been a big part of my education as well. We started working with Hybrix Manufacturing 3 years ago. We met Greg, I think, 4 winters ago. And I don't say this to fluff your skirt, Cindy, but this is my favorite meeting. I get to go meet a lot of people, and this is by far my favorite meeting. What you guys are doing here is unique. We work with a lot of, commercial ag people. This is different, and I can't encourage you enough to really take advantage of all the great people that are here and how you share ideas. Okay, moving on. Otherwise, we're going to be here all day. So Vic and I are going to talk, not in tandem, but he's going to follow me. Are you guys hearing some feedback? Does that help? Greg and Cindy wanted me to cover humic substance. So last year I did a little talk on this. How many people here in the room apply humic acid to your crop, either in the seed row or throughout? Okay. Who used humic acid 10 years ago? Who used humic acid 15 years ago? I win. Agronomists brought humic acid to Riddell Farms in 2010, 12. And I've got a story around that. But humic acids, all I want to do today is spend a little time, talk about what humic substance is, what humic acids are. That's not my friend. That's me, blah, blah, blah. I love talking about myself, so we can keep going on that if you want to. So I'm going to be reviewing humic substances. Then Vic's going to come in and we're going to talk about the keys to success, about product quality and actual humic product quality, redox, humic and fulvic effect. This talk can get as deep as you want depending on your questions. More from him, not from me, okay? I'm just going to be fairly simple. I want to answer these questions. So what are humic acids? How are they defined? How do we measure and identify organic matter? I want to step aside and talk about that because that's part of this humic molecule thing. Why do we care? What are humic products made from? And then what humic substances are not, because there's lots of confusion out there in the world about humic. What is it? And what will it do? So let's read this together. Humic substances are relatively large, complex, mostly carbon molecules that function in the soil system as concentrated organic matter and are stable across a wide pH range. That'll come in later when we talk about standard testing or evaluating. Humic substances are the dark-colored substances that remain after the natural biodegradation of biomatter and whose distinct characteristics are their relative resistance to further biodegradation and are highly heterogeneous in their molecular structure. What did I just say? Does that help anyone at all? That's why we're here. So let's answer this first question. Where am I getting the feedback from? Does anyone know? This guy. Oh. Smart guy. Good job. Thanks, Jeff. I met Jeff from coming back from the airport. So first, let's think about humic substances in nature. So question, do humic substances form in living soil without inputs? The answer is yes. We got a head nod over there. So the black, heavy soil, the black color we see in soil that is stable over a wide pH range and is resistant to further degradation is defined as humic substance. That is true. So in a living, I just wanted to start there with living soil. In a living, biologically active soil, you should be and are creating humic substance, okay? They're generally created in that top 6 inches of soil. Why? Because that's where the biological activity is. Humic substances can be degraded. They can be oxidized, excessive tillage. They can be degraded by anaerobic conditions. And this one is really important to know, that humic substance is degraded by all forms of synthetic nitrogens, okay? There's actually a lab procedure if you want to dissolve a humic acid or a humic substance. You treat a known amount of humic with a concentrated solution of urea. It will dissolve all the bonds that make that molecule stable. Is there any surprise that we've lost the amount of black soil in our modern chemical farming system? I came out of the chemical farming world, and it breaks my heart to think about, like Lauren said earlier, where would that farm be today if I understood some of this stuff, you know? Is anyone else in that camp with me? You wish you would have known a little more? One more way to describe humic substance is they're the natural organic matter that has become stable and complex by the process of humification. Again, I just said nothing. We'll talk about humification here in a minute. That's the pointer. And I don't want to go backwards. Okay, we're going to step back just a little bit and talk about organic matter. So, Midwest Labs soil test. There's a number right there. Does anyone know how they come to a number of 3.3% organic matter? Does anyone know the name of the test, the process that they do to get to that number? If I was Glenn, I'd have a pocket full of dollar bills and pay you right now for the right answer. Okay, so zoom in on this guy, organic matter, and it lists it right there. It says LOI. So what that means is soil test, 0 to 6 inches, dried, then baked at over 600 degrees Celsius. Burn out anything that's flammable, okay? The difference between that and the original weight of the soil is by weight a 3.3% organic matter content. Does that help us? First of all, are high organic matter soils better than low organic matter soils? Nodding of heads. I would agree. These are the dark black soils that we all think of when we think of healthy, fertile soil. But what does this number mean? There's the oven, and there's the calculation. So again, what does it mean? Loss on ignition does not describe the organic matter in natural soil systems. Let's talk about this pie chart for a minute. About 1% of this type of testing, or truly organic matter, is living organisms. So these are the microbes and the macro fauna in a soil test. 10% of that burnable material is green plant material, so it's roots generally. This portion, not that, this portion would be any of your crop residues. So these are highly lignified materials. They're going to require, I'm going to stay away from here. They're going to require decomposers, okay? These are your saprophytic fungi, your decomposing bacteria. It's this portion of organic matter that matters for us to build a complex carbon that is resilient and can exchange cations and do all of the magical things that a biological system needs. The rest of it, the LOI doesn't really tell us about that. So the description of how humic substance is formed, and this is simple, these resilient humic type carbons are formed, assembled in a living soil system only by the interaction of root exudates, sugars and enzymes produced by the plant, consumed by the microorganisms to form humic substances. So these things are formed in living soil, but they're also found in nature in deposits. So, and we'll get to that in a minute. So how I just defined humic substance right there is very simple and crude. That these extremely complex and resilient carbon molecules are formed by the interaction of root exudates and biology. Okay, here's how and you can laugh at me if you think this analogy is silly. Picture building a home with 1,000 different groups of tradesmen. Okay, you with me? You've got framers, you've got concrete guys, you've got, the plumber is there. Every craftsman, tradesman that you can think of are working to build a home simultaneously and for a very specific reason. And this home is all one building, okay? You with me? This is a big molecule, and we'll talk a little bit more about that, but it's a large, complex, stable molecule. And even saying that is oversimplifying it. Okay, so humic substance is a product of microbial process, the interaction between plant roots, exudates, microbial, and the microbiome. Okay, plants will pump 30 to 50% of sugars into the soil. And we know why it's there. We know why plants do that, because they're not only trying to build stable carbon, but they're getting a direct benefit from the microbiome. Our philosophy at BioAg, and we build products with this in mind, is absolutely the same as how Back to Your Roots and Hybrix Manufacturing view the priorities of soil. Air, so we need an aerobic system that can respirate. We need a place for biology and support for biology, and then build complex carbon. We absolutely think in those terms. And just the reason why I mention that is because truly, and Glenn and everyone here that's going to speak, or Greg and Cindy, they're all going to say that in the end, this is a priority. And it's the soil system that can build this. Okay, let's look at, now this is, sorry guys, I struggle with pointer. So we're not going to spend a lot of time here, but I just wanted to make this statement that with our ability to test and measure carbon groups in particular, they've found over 800, we know, and there's way more than 800 organic compounds from 6 specific functional groups in humic substance. Okay, that doesn't mean that all 800 organic compounds are present in every humate, but it does mean they are present. They have found them in humic substance. So this is a very small molecule structure of what's possibly in humic substance. Okay, and we know that because they can measure, those hydroxyl groups, the total carbon. But this is a guess, a pretty solid guess from the organic chemistry world, of kind of the beginning of a stable humic structure. You can see there are so many, and I get this slide from a guy I really respect. His name's Lawrence Mayhew. He's been researched. He's actually one of the founding members of the Humic Products Trade Association. He has studied humates his whole career. He's now 60, mid-60s. Cindy, you and Greg didn't get to listen to him talk at Acres, did you? It's fascinating, fascinating. And he is an organic chemist. I am not. But he mentioned that all of these R groups in this structure that I think he put this together because he could identify many of these things. He said all of the R groups in this structure could be something completely different, including mineral and different groups. So in humic structure, there is no one definition. It is absolutely heterogeneous. No one can define humic substance as that molecule. And that's important to know because as we move along here, we're going to talk about why humics can vary, as particularly humic products. So why should we care about humic materials? They promote healthy soil and microbial populations and diversity. They enhance nutrient uptake by plants. They have very high water holding capacity. They have extremely high cation exchange capacity. And for those of you who don't really know that term, it's just the ability of a substance or a soil to move cations and trade them, which is really important in a biological soil. Extremely high CEC. They have both positive and negative chemistry in the same molecule, in the same structure. And they can complex cations, and they also stabilize anions. They will stabilize phosphorus and nitrates. They have a very high buffering capacity. So like all things found in nature, humic substance is averse to big pH swings. So if you want to change the pH of a humic substance, you have to manipulate it to the extreme. Natural products, natural substances always want to settle back into a biologically friendly range, and that is true of humic substance. They support natural plant defenses against stress directly. So in a minute, we'll get to the fulvic fraction. So who here knows about what is known as fulvic acid? Show of hands, fulvic acid. So that technically is not an independent acid from humate. Fulvic is the fulvic fraction of humic acids. Okay, you with me? It doesn't technically have its own definition. You can identify it, but it is the small molecular weight portion of humic substance. If my Dodge pickup, 3/4 ton, long bed, if that is a humic molecule, I'm trying to give you a little scale, a ballpark analogy would be the fulvic molecule would be the size of my steering wheel. Are you with me? Fulvic can move into plant cells directly through the roots or the stomata and the leaves. It's a small carbon, and it is soluble at acid pH and alkaline pH. Okay, it's a very resilient molecule. So let's talk about product. How am I doing on time? I'm 27. Okay. Let's talk about products built with humic substance. So I'm going to tell this little story. I raised my hand because I was the first one here to try humic product on the farm. Agronomist shows up with 2.5 gallon jugs and says, put it in the liquid kit on the plant. We're going to do a test. and we probably ended up purchasing the product, but we planted most of the field, thankfully, with the product. It was a gallon rate in the row. So we were planting tall fescue, turf type tall fescue in the spring, and in the Willamette Valley, that's always a tricky deal. So our farm, we grow seed crops. So the Willamette Valley is the grass seed capital of the world. Turf type and forage type fescues, perennial ryegrass, annual ryegrass. Turf type fescue, plant in the spring generally, and we don't have irrigation, so you try and get it in as early as possible. You hope that it survives the summer because our rain shuts off the 1st of June, and we won't see rain again until October, okay? So wet soils, wet winters, wet soils in the ground, but planting tall fescue in the spring is always a roll of the dice, right? Some years you hit that rain in June and that crop makes it. We also get really hot. So we can get, hit hundreds 3-4 days a year. So it's always tricky. Well, this one year we planted this tall fescue crop, 20-inch rows, treat, untreat. We just we skipped a portion. And we thought we had found the miracle, right? The humic substance planted, the skipped row, or we skipped a chunk. It was a pretty good test. It struggled. First of all, it didn't tiller like the humic treated stuff did. And we get into August, September when it's hot. And the humic treated fescue looked wonderful. And we're like, we found the magic. Here it is. It's that one ingredient that can save the farm kind of thing. We did the same thing next year with a different product. And guess what happened? Did we win again? Yes. Epic failure. That product, and it was a different product, actually hurt that crop. And we didn't get the stand. And we didn't know why. With the time I've spent on humic substance, working with Dr. Faust and these experts and talking about it, now I know why. 2 really important reasons. No, my slide went away. There are 2 really important reasons why humic products can have very different effects on a crop. One is the source material. So Leonardite mines, humic shale, whatever they mine to build these products can have very, very different starting points. Guys, I struggle with the clicker. And the source material, where these products are, and what they're made of coming right out of the ground. So this is a picture of kind of I think it's not a mocked up, it's a real slide of a humic deposit. So some sort of overburden, usually a clay stringer, clay layer, and then that's where they find this material called leonardite. Okay, you can build humic substances, products out of leonardite. You can build them out of peat materials. You can build them out of compost materials, composted materials. Any organic substance that's been through the process of humification. Okay, why are most of the commercial products on the market built out of this substance? Anyone? It's concentrated, very concentrated. So these products can have up to like 60% soluble humics in them. Whereas a peat material might have 10, 12%. Compost might have, 5 to 8%. I'm really curious about Brian's compost. I'm sure there's a way we could find the fulvic fraction and the humic fraction in that product. But that's why these products are built out of lignardite or humic shell. Don't take it away. Put one more. So here's a peat bog, and here's a mine. So I said there's two really critical things that determine the beneficial, the benefit of a humic product. One is the source material, and two is how it's processed. Okay, so there are lots of chemical processes out there, and there are very sterile humates out there because harsh chemistry is used. They change the molecule with chemistry. It's easy, quick and dirty. Lots of chemistry, swing the pH way high and then dump acid in, and that drops out the humates. But it's harsh chemistry. It's different. So where the substance comes from and how it's processed. Very important. That description that I just said, those two factors of the value of a humic substance, I believe, describe what happened to us at Riddell Farms in 2012. We had a product on the farm that was wonderful and beneficial. It was the magic. Then the one we tried the next year, it was dead. So keep that in mind as producers, guys. And I know you don't have time to sit and study every humic substance or every product you're applying to farm. So just take my word for it. You can ask some of those questions around how things are processed and get an answer. And it's important. It's very important.
So where the humic is sourced, like say Alberta or whatever states, is there places where you don't get it from or do?
We are very particular about where our source material comes from because we build full power. Okay, we need a high fulvic fraction material in order to extract the fulvic fraction, okay? There are mines that they may have a high humic test number, but there is little or no fulvic fraction in that. You with me? So yes, the mines are very, very unique, and Vic can cover more of that. And I know where a couple of the mines that we use, but he has sent truckloads away that didn't test. So they weren't what they were supposed to be. They went back. We didn't buy them. We turned them away because we have to have a certain quality to build what we build. So I'm just going to mention a couple of these things and then I'll be done here. Want to talk about what humics, hang on, I got to talk about extraction for just a second, or processing. So I like these quotes. I think I made that first one up. Considered a multifaceted organic magnet, and right now I'm describing humic substance as a biostimulant, and I'll tell you why in a second. They're considered a multifaceted organic magnet, functions in the rhizosphere as a buffer, a bridge, a conduit between inorganics, clays, minerals, micronutrients, microelements, components in the soil system, and the living organic microbiome. We've already talked about the 800 or more organic compounds. And I just lost my train of thought. And I don't think my notes cover that. anyway, so yeah, let's talk about the fulvic fraction. So I mentioned Lawrence Mayhew, who's been working with humic substance and trying to define and has defined through the Humic Products Trade Association standardized testing to evaluate these materials. Simple thought, think of heavy humate products as oils, a crude oil will, um, or what have you, and the fulvic fraction as gasoline. So it's been refined and separated. That's just kind of an example of humic substance and what the fulvic fraction looks like. So this is Lawrence Mayhew's slide, and we added something to it because he described the extraction of the fulvic fraction in this same manner. So they swing the pH up, whatever's soluble at a high pH, then they treat it with an acid which drops the heavy stuff out, and then they have the fulvic fraction. So that's a standardized test. We don't use this process. So, secret material or process, but we actually use a biological process, and it involves organic materials. But we don't do this. We don't use chemistry and swing the pH big. So one thing that the Humic Products Trade Association did was they found or developed a standardized, repeatable process to define the humic fraction. So if they have a material that has a humic claim, or sorry, a fulvic fraction claim, they have a standardized test. It's called this ISO test method, and there's a number on it. We actually have that test in our lab, but there's only a handful of labs in the country that will test for this. How many people here have seen a fulvic acid material that claimed something more than 5% active ingredient? Anyone? Because they are out there on the market. And Lawrence Mayhew will tell you, after all the materials he's tested, after all the mines he's worked with, he said he has not been able to find a humic or a fulvic fraction product that will test above 5%. And that's just because, once again, natural products are not concentrated things. They are balanced things, okay? Kind of a simple slide about size and scale. I think my analogy of my Dodge pickup versus the steering wheel is a little better than this, because stable humic substance is in fact very large. These are large, complex molecules. This one is another reason why there are different plant responses and experiences with humic substance. This was a study done quite a long time ago. But if you look here, I'm going to get down here so I can read these numbers. So these were humic substances, Leonardites, that were tested in a lab in Russia. If you look at the contents of ashes in the initial raw material, you'll see that this one right here was only an 8 to 10% ash. What does that mean? That means that 90 to 92% of this material burned. With me? This guy here, he's in the, so he was be 69%. No, I got to do math. 71%-ish burned. So these were the ash portion. These are the portions that did not burn. So therefore, the contents of the humate in this product was 70 to 80%. I didn't even have to do the math. It's right there. If you follow this through, so these are bioactive groups, ketones, carboxyl groups, phenolic groups, the aromatics. These are all biology drivers. And the end result here, this material, the top line, 83.8% of it was biologically active, okay? This is a very rich material. The others are fairly similar. They have, they differ in a couple different things. Some of these different groups are different. This one is clearly the most concentrated organic humic substance. If you look down here, it says they did trials and it showed the crop capacity of potatoes and oats increased by 22 and 61%, and that was with this Irkutsk humate. Particularly good results were obtained when testing cucumbers and carrots with a crop capacity increase of 80% and 60%. Okay, so once again, source material matters. And in this case, no surprise, biologically active portion, higher biologically active portion produces more biology, therefore plant production. Okay, I just remembered what I was going to say. So you will hear claims about humates, and I'm going to close with this, and Vic's probably ready to jump in anytime. What humic substances are not, okay? If someone ever tells you that an application of a humic product will increase your soil's CEC, that's not true. Because you think about it, yes, these are carbon rich, these are carbon dense, biologically friendly carbons that we're applying to our fields. But if you crunch the numbers on the amount of carbon that's already in your soil in the top 6 inches, it's, tons and tons. It's 80,000 pounds per acre. How are you going to swing, number one, the organic matter level or the CEC of a soil with this tiny little application? You're not. Not directly. Humic substance applications, particularly in ways like in the seed row, near seed, will change CECs, will change organic matters over time because they're priming the pump. We're not adding a percentage of carbon to our soil. We'll never be able to track that on a soil test. But what they do is they help drive biology, sugars, and here's the magic cycle. Microbes, photosynthate, root exudate, biology carbon, more photosynthate, root exudate, biology carbon. That is the cycle, and you can drive it faster and faster, and that's where these types of products. Hi, Vic. How you doing? He says 5 minutes, whatever. And one more thing, and I'll finish with this. If you ever see a product that says it's derived from plant material, or it's not you can figure out that it's not defined from a humified substance, it's not humic acids. And that's another one of those things out there. So if you ever find, and the only reason I'm talking about this is because Lawrence Mayhew deals with this all the time, and he tests these products that are on the market that are not in fact what they say they are. And he hasn't made a career out of it, but it's a big part of what he's done. One of the big violators out there, so beware, are products built with lignosulfonates. So these are byproducts of wood processing. If you put them in a colorimetric test against fulvic acid, they will appear to be fulvic acids. And there's only one way to figure out that they're not, and that has more to do with their sulfur content. The fulvic fraction of humic substance will never have a big sulfur number, even a measurable sulfur number. But lignosulfonates will. And so it's one of those, kind of beware things out there around the fulvic fraction. He had one more slide up here. And this is just this was a fun treat, untreat. And something to land on here is the earthworm population. This was a fun trial because this fall planted, it was a tetraploid annual ryegrass. It was fall planted, and this entire field received 80 units of nitrogen in the fall. Now we get a lot of growth. We get a lot of good growth weather in the fall, so we'll plant, and a lot of times plants will be up big and strong. For whatever reason, they chose to throw 80 units of nitrogen on this. It all got 80 units of nitrogen, and that's what the humic product did in the seed row. It's really exciting. Okay, guess we can do some questions. We're going to head toward this talk. And that would be with Mr. Vic Sakhalkar. I'll let you introduce yourself.
Question? Question?
Listening.
What's the difference between biochar and humic acid?
I am so glad that Vic stepped up here right now. I can answer this, guys, but I'd rather that he does. I really would, just because.
I thought people are going to fall asleep with Bill's smooth jazz voice and lunch. But because it's so late, that's good.
I'm going to say a quick answer. Where'd that question come from? Where are we?
Anonymous.
Greg, was that your voice? Oh.
Where do you put this? Can you guys hear me? Right here?
Good. Okay. So the quick and dirty answer, is this working? The quick and dirty answer that I'm going to give you, and you'll follow up, is biochar is in fact carbon, but it's dead. Can't hear you.
Biochar is a carbon. Sorry.
Wow.
Biochar is a carbon, but it's dead. You want this back?
No.
So it's too loud. Did I wake you guys up? Is it better? Thank you, Jeff. It's a carbon cycle, right? It starts with food, goes to compost, goes to peat bogs, and then goes 10,000 years later, it becomes lignite. Then it becomes brown coal, then black coal, then diamond, right? So it's a carbon cycle. What you have in a biochar is simply take that initial carbon, burn it without oxygen, and you get carbon. So it has no functional groups associated with it. It's carbon, right? So it's just a complete different chemistry. The humic substances are, you got to catch them at the right time in that timeline to make sure that they're bioactive. Does that make sense? Thank you. So let me, so just come here. A little bit about, I think it's a little important that when we talk about humic substances, and particularly in your soil system where you're reliant on very little input, just a few gallons to cover lots of acres, it's important to understand why the small amounts of products drive bigger changes, right? And it's really important for us to share that how the products are built. And it's really important because that is the key, that good quality product, well-built product can drive those changes. So I'm going to cover a few things. Bill covered some of the theory. I'll get into the quality and what we do, why we do it. Then we get into a little bit about understanding what happens at the root and how the humic cycles drive the function of the biology forward. And then we'll take the Q&A. So the getting into product quality, right? So a little bit about company information. I think Bill covered some of that, so I'm going to skip through some of these slides. But it was established in February of 1981. So we'll be 45 years company this February on the 20th. We have experience in sourcing, old line company. I'll cover some of that. Bill covers some of these topics as well. Acknowledgement to the company founded by Dr. Robert Faust, a lifelong scientist, deep interest in ancient history and agroecology, and pioneer in development of humic acids, mushroom-based supplements for human and animal nutrition. I purchased the company from him in 2020. That's a little bit about me. I'm a technologist. And so I built some cars before. I built Intel chips for 8 years after that, and I'm building ag products since 2012. I have 2 companies in India, make amino acid manufacturing and azadirachtin manufacturing, and then BioAg. And I have a 4-year-old and 12-year-old, live in Monmouth, Oregon. I say I'm a technologist because I love reading books to my 4-year-old, and she doesn't go to sleep. I pull up a book about silicon, and she falls asleep. That's my reading material nowadays. That's our team. This is a little slow. Okay, got it. Family company. Okay, so quality is, when you think about quality, is it a defect reduction or is it more than that? And so I get to bring a lot of the concept of, I work quite a bit with the Japanese manufacturing principles, and I bring some of that to the manufacturing side, and I'll explain why does it matter. We cover quality at every step of the way. We cover quality at the customer value. We love getting some of the feedbacks from you guys about how our products are working in the field. It's called Kano. Process quality, we have a process quality check every step of the way. We check all of our products are filtered, 80 mesh minimum, and we do check at every batch that goes out of the factory. Process quality, product quality. I'm not going to get into details of this for the time's sake, but we do, Bill can attest to this, we have about 20 versions of our product because we go and do a quick check. We see, we get the feedback, we make an upgraded version, and so we've been constantly doing this plan, do, check, act. I'm going to skip this for the sake of time, but let's cover this. Okay, so um, manufacturing process, ingredient, guarantees, those are the core categories. So for fulvic, we have a no chemical extraction of fulvic isolate. That's the manufacturing process. Ingredient, we have multiple sources of the raw ingredient we bring in, and we guarantee what they call hydrophobic fulvic and total fulvic of the product. These are ISO test method. We also guarantee acidity and carboxylic and hydroxyl group of our products. Why? Because there are a lot of products out there that claim to be fulvic and humic, and we tend to make sure that our products hit those quality numbers. Humic, the same way. We have a one-step conversion from insoluble to soluble. Our products are not extremely dry. They're very bioactive. We use a CDFA, California Department of Ag test method, that eliminates ash, human, and gives you the exact only total humic acid number. Multiple sources of linodite and peat as a source. Aminos, Bill will cover a little bit about aminos. Aminos are manufactured in India as part of the soy protein hydrolysate, and they come from non-GMO soy flour. And the guarantees need to change. We cover guarantees for every amino group that Bill will cover later on. And then we have we manufacture silica as well. So I want to cover this topic a little bit, right? How many of you guys know redox? Or have you heard of redox? Okay. Well, I mean, the redox is just simply stands for reduction and go up. Their company named Redox, and some of the names are coming up as Redox. It's really simply combination of word reduction and oxidation. Okay, that's what redox is. Why does it matter? I can explain humic. Why does it work in the small amounts much better with redox principles than the pH principles? So we'll cover a little bit of this. Humic acid and soil redox, microbial processes, redox and stress, humic acid, and redox buffer. So I gotta walk around. I can't stand at one spot. So when you think about, can you still hear me? When you think about pH, it's a potential of hydrogen, right? We covered that earlier, and that is ability to exchange hydrogen, right? So what it means is H2O breaks into H and OH, and H form H3O, which is a hydronium ion in the soil system. If H3O is greater than OH, the system is acidic. It's a low pH. If OH is greater than H3O, the system is basic. And that's what the entire university and science knew since 1960s. Redox ideas are coming in later on. They started about in 2000. And that is not about hydrogen. It's related to hydrogen, but that is about exchange of electrons. Okay, so in a soil system, just think about this. I'll walk this example throughout the presentation. When you think about a red-looking oxidized soil, right, chances are it has some rust in it. So that's an iron, Fe3, 3+++, right? When you start working with microbiology and we start bringing some of the good products in, it has a tendency to reduce the iron from Fe3+ to a soluble green iron, Fe2+. And that's not transmutation, that's not magic, right? That's just simply electron exchange. It's moving from Fe3 plus to Fe2 plus. And that's when, so any question on redox? Does that make sense? Okay, my voice is also very smooth, and people are falling asleep during the coffee break. Well, I'll continue. If there's a question, raise your hands. Okay, so you guys know about pH, right? Our pH has a specific range. It's important to have the pH soil system in certain range, but it's also important to have a redox of 400 to 450 millivolts in the soil system for it to function really well, right? That is an oxygen-rich soil. You can have oxidized soil, and you can have a super really reduced soils, right? Give you an example of oxidized soil. It's a dry soil, which is, it looks red, yellow, dry soil, high pH, right? And the very really reduced soil conditions are bogs, where you can smell H2S or methane or hydrogen sulfide, right? So those are extreme conditions. But it's important for microbial function to occur is these two functions, not only pH, but also redox. The ability for that system to exchange electron has to be there. Now, it's a little difficult to test it in a soil system, and that's why it's not so popular. You can test it, there are test methods available, but you need a silver electrode and platinum electrode and gotta find it right space in the soil to check the conductivity of the electron conductivity between the two electrodes. You can do it much better in a liquid format, right? So we try to get our redox potential for our fulvic right. But that is the concept, right? You have to have a pH and EH in the right balance to get your soil to function really well. So redox is a reduction in oxidation. In soil, it refers to transfer of electrons between substances. Okay, could explain a little bit about those electrical impulses that we're trying to understand. High redox means high oxygen. It can be excessive aerobic soil, right? It's oxidized soil. That means it has low electrons in it. Low redox, that means low oxygen, that means anaerobic. Why is that important? Because it links to what humic substances, good quality humic substances can do at the root zone. So it's a key indicator of soil aeration, crucial for understanding a wetland and oxidized soil. Ideal soil redox is between 400 and 450 millivolts. Okay, so taking a step back. When you have an organic carbon present in the soil system, that with microbial processes that includes water, combine those two things releases CO2. Glenn covered that, we know that, right? But it also releases the free electron, and it has tendency to reduce the environment. That's why plants can function in oxidized soil. It happens at the root zone. So this is pretty standard process, right? Everything is going smooth, but then the humic acid becomes important because it provides that buffer. It provides that electron exchange. That's a big molecule, organic molecule, that provides the exchange when there are stresses in the system. So when you have highly oxidized soil, highly anaerobic soil, drought, heavy metal, when the stresses exist, humic acid can help. That's a fundamental way to explain humic acid. It also does pH buffer, right? Like Bill was pointing out, that you have a humic substance, it's a liquid, you try to move it, pH, add citric acid, and this and that, it won't move very easily. But it also has that buffer, which, that has ability to exchange electrons. So that's what humic acid primarily do. So In an oxidized condition, when you have excess oxygen, use humic acid as a donor to transform electrons for contaminants. In anaerobic condition, microorganisms utilize the humic acid as a terminal acceptor. So I'll explain this a little bit later. Essentially, it's a nutrient cycling. It's a tool for providing nutrient cycling, right? Example is of these things is contamination reduction. And you see this, you can test this, and it's proven over and over again since 2000s. But when you have soil organic carbon, highly available, good organic carbon, it'll start reducing the Fe3 to Fe2, Mn4 to Mn. So it'll start moving those things in the right direction. It'll start making minerals solubilize at the root tip. That's what it means. Okay? And you can explain that much better with the redox principles. Okay, so why does it matter and how can we test it, right? Because the tools are not there. The EC doesn't check for it. EC checks for your pH. EC is essentially two electrodes, right, that you pass alternative current between them, and you say how many cations are moving between those two electrodes, and you check that. That's EC. You can't check EH very easily because it's free electrons, right? But the way you can check it is you start seeing earthworm population when you apply these products. Why? Because when you combine high quality humic at the root tip, and I'm just going to go back to the oxidized soil condition, right? Oxidized, everything is oxidized. You got 10,000 thousands acres of field that's oxidized, and you're looking at pH 8, 8.5. Things start as soon as the plant go in. We all know that. At the root tip, the microbes start functioning. They start solubilizing minerals, right? Humic acid, if present, acts like a battery for that. It provides that exchange for it to function more effectively. Now, in the soil system, you also have cocoons of the earthworm. Plentiful available. They get spread by animals, birds, they're there. But you don't see a whole lot of activity in the dry, arid soil, do you? This is consistent response. Who has applied our products and start seeing earthworm population pop up? They start coming in because you're giving them micro homes to be able to propagate themselves, right? So it's really key that when you have organic matter that is not, and need to have a low oxygen relatively, right? Super oxidized soil, you won't see earthworm. So you gotta have relatively low oxygen and no anaerobic condition, and then you will start seeing some of the stresses go down, and then you start seeing the earthworm population go up. And earthworm acts like a microbial multiplier. And that is the key. That's why you can use small amount of humic, small amount of fulvic right next to the root and change the system. Otherwise, you have to use a lot, right? If you think from a pH standpoint, you got to use a lot to shift. But when you can combine that with microbial function, you're letting the system do the work. Okay. So redox balance is important for functioning soil. High quality humate, important, provide redox buffer to microbial processes. When the humates are not high quality, they tend to burn the microbes out. They will dry them out. They won't produce the electron exchange that's needed to trigger the process. So they can work in the even small amount. So, okay, that's humic for soil. Thank you for listening to me so far, and I can kind of see your eyes are still there. It's good. So now we're looking at plant redox. So we're talking about stresses, right? So the redox explains stress. right? This is my fourth grade biology book. If sunlight, CO2 comes in, and you release the O2, water, and sugars, right? A little bit later, it's my slightly improved version, is that this is a cytoplast, your water coming in, you have chloroplast, inside chloroplast, light energy comes in and you produce O2, CO2 comes in, there's a thing called Calvin cycle, and I'm not qualified to talk about it. I'm not PhD, right? I just talk to research papers with my 4-year-old. So that Calvin cycle is pretty complicated. But what's important is that one produces the glucose. And within the system, there is an electron exchange inside a cell. So soil system, I talked about, now I'm going into the plant, right? And we'll talk about that with respect to fulvic. So NADP is an oxidizer, NADHP is a reducer, ATP, you've heard about that, ATP for human health, et cetera, That is a adenosine triphosphate. That is a currency to see if the process is in balance or not. And then ADP is a counteract. So electron exchange are happening inside of the cytoplasm as well, okay? When stress occurs, okay, so everything is running smooth, plants are happy, they're being well fed, no problem, right? Tropical plant mostly. When the stress occur, biotic or abiotic, move closer, things go wrong and electron accepting donating process goes off. So what are the stresses and what are those chemicals that are formed, okay? So stresses, you guys know, the air pollutants, drought, high light intensity, salinity, right? All of those things. They will generate what they call it ROS, which is a reactive oxygen species. And it's nothing. Hydrogen peroxide. How many of you guys know hydrogen peroxide? That's just a really high reactive oxygen species that in a small amount, plants will produce it for signaling, but when there are stresses, then they overdo it and then kill the cell, right? So anything which is O minus, O2 minus, H2O2 and OH, those are radicals that get produced inside of the cell. Not so much different than a human cell, by the way. Similar processes happen in human cell. We, of course, don't take the light, but similar processes happen. We also have ATP function, right? That's why some of our fulvic acid also goes in human health. So it's called reactive oxygen species. They are strong oxidizers, and they'll kill the cell. That's when the fulvic acid comes in. Now, fulvic acid, if you remember the molecule, it has an electron exchange capacity, right? So it'll come in, it's a strong antioxidant, and it will reduce the ROS. So it can donate, accept electrons, and it'll help ultimately in production of the sugars. Okay, so I'm not going to read through this. It's just for your reference why ROS gets reduced and how good quality fulvic acid, which is not adulterated with hydrochloric acid, sulfuric acid, is biologically derived. It's very clean, will aid in this function. And here's a simple example. It's an isolated example. It's a 21-day lettuce, harmony lettuce trial done by Dr. Conjoyan, well-known researchers in the CEA space. In the top part, he cut the water pump down, right? It's a NFT, so it's a nutrient film technique, just water running through the system and nutrient running through the system. And he cut the pump out for 12 hours, right? And then fed with the fulvic rate 0 to 10, 30 ml per gallon. 30 ml is about 1%, right? You can see clearly that drought stress and fulvic response, it helps, right? When there's no stress, increasing rate will also help. right? There's an optimal rate where it will stop because plants have what they need. So fulvic will reduce the reactive oxygen species in the cytoplast. So in summary, humic substances are soluble organic matter, right? Humic for soil, fulvic for soil and plant, and they provide redox buffer. They provide pH buffer too, right? But they also importantly provide redox buffer for soil and plant in conjunction with microbes. That's why you can apply small amounts right next to the root and the entire system functions. Okay, we also have the micros that we add to it, and makes it more efficiency in terms of how many people heard rhizophagy cycle? So a few of you. So plants, newer concept, right, is that plants harvest microbes. They take the microbes in, they eat them. Essentially, rhizo is plant, phagy is eating. They eat them, and then they oxidize some of them in the form of new root hair development. So when you add some of the microbes, including cobalt and moly, it helps with the rhizophagy cycle. Okay, so one of the thing I want to hear from you guys, I think Bill asked this question, how many people used our product? And I use some of you guys. Thank you. Any feedback, please let us know, right? Any experience with our product, let us know. And I'm available for question and answer. Bill will cover the aminos after this. There was a request to cover amino acids.
Come on, someone's got to have questions for the day.
Question?
So you maybe tell something about the extracting, like the local cargo. What is that? Humic, is that called?
Humic. Yeah.
So there's a humic acid is extracted with potassium.
Yeah.
Can you tell maybe the difference?
Yeah.
You can repeat it.
Yeah, I can repeat it. So the humic acid, the question was, is humic acid extracted with potassium? And yes, there's truth to it, right? So the humic acid, when you bring the raw, just an example, the raw linardite comes in, it has a 60% humic acid, but that's not readily available, right? So in order to solubilize it, you can use ammonia, sodium hydroxide, potassium hydroxide, anything which is a high pH, you can add to it, right? Problem is what I've seen in the field, you use excessive amount because people start making potassium claims, right? So we start using excessive amount of that and not to. What we do is we start that process, we trigger that process and let biology finish it up. But you have to start with some potassium hydroxide to that process. Now, fulvic is different for us. It's very clean. We use more, you can think about this more like a wine manufacturing process, right? So we bring some microbes in, which consume the matrix. They break it down. Then we use enzyme to extract it. And then what's left over, we have an original, it's kind of like making yogurt. We have original material that we combine with this material, and then we extract fulvic from it. So there's no chemical added in it at all. So you can overdo on hydroxides, and it can really, what it does, it damages, it won't let the pH cycle or redox cycle or microbial cycle work as efficiently when you have so many chemicals added to it. Did I answer your question?
Okay. So what happened with the human? Like you talked to humic and fulvic?
Yeah.
What happened with the human?
Human?
Yeah.
Yeah, we take it out. We don't. We actually So the fulvic process. Well, the human and Olmec and some of the other things that are part of the humic manufacturing process, right? Mm-hmm. So we have to take some portions out. So generally, depending upon mine and where you bring the product from, it shows up as like a waxy coat and a really waxy, thick substance, that's what you take it out. It's insoluble. And then our final product doesn't have that. Yeah, it's still, I mean, it's still, it still helps with the soil structures and things like that. So we can blend it in some of our dry products to get it out on hazelnuts and things like that. But it doesn't go into the finished product.
And it's just a small portion then.
Human is a small, insoluble portion. Yeah. Yeah.
So is the fulvic the best use in the foliar or on the root?
I think it depends, right? So what I would say is it depends on how your grow system is. So if I'm talking to a blueberry guy, right, it's a high input, right? And when you have foliage, it has ability to take the fulvic in, and you can see that when the fulvic goes in, all the other things come in. Calcium comes in, silicon comes in, boron comes in, right? It kind of has that entourage carrier effect. But when you're using very little to trigger the plant growth, I would suggest to use it as part of your blend, along with the root. If you're going with 1, 2, 3 gallons per acre, but you can always use fulvic as part of your foliar application later on to reduce the stress. Because what fulvic will do, especially if you go with nitrogen, right? Nitrogen, ammonium nitrate, whatever, that stress will be reduced. And you can see it. You can see the clear line where you apply fulvic and where you don't. It'll reduce that stress. Good question. Anything else? Yeah? Question? Oh, you're stretching. Okay. All right. Where's Bill?
We're gonna take a 7-minute break.
A little background here. We've been working with soy hydrolysate, non-GMO soy hydrolysate fertilizers for a number of years now. The products are built, and I say this just to give you kind of the scope of all the fun things I've gotten to play with over the last few years. The products are built in India. They come to us they're dry products, so they come in 50-pound bags. And they range from just a straight, what we call a nitro-amino product, that is a 1400 on the test. And then there's a whole range of products that are complexed with micronutrients, including one called multi-amino. It's got 7 micronutrients in it. So I, along with all my work with humate applications and sampling, soil sampling, sap sampling, tissue sampling, I've worked with these materials as well. And they are organic materials, if you can get them registered. And so I'm probably going to tempt y'all, growers, particularly the organic ones, with products that you don't have access to yet. But we're working on that. Show of hands, who here has heard of or used an amino acid fertilizer? Was that, for those that raised their hands, and you just yell at me, were those products that were a soy hydrolysate? Were they a fish hydrolysate? Both? Okay, so you've got some experience with these. And we'll talk about, you've heard, if you work with them, you've heard about them, that they're highly efficient forms of fertility for plants. As you can imagine, the soil microbiome loves to consume them, and they are also available to the plant foliarly. In the case of soy hydrolysates, you're basically feeding an amino acid profile from a plant to a plant. Okay, so generally the amino acid need is kind of matches what a plant wants to have in its system. Oh, here we go again. Wrong way. Okay, so amino acids, something we probably all have heard in a reasonable education system is that amino acids are a precursor or a building block for proteins, right? They're also a precursor building block of enzymes, which are very important in how bodies function, how plants function. But here's an example, or here's a little description. So right here, we have a chlorophyll molecule. So that's like a very basic description of the chlorophyll. And here are these amine groups connected to what? Magnesium, okay? Similarly, here's a picture of a hemoglobin molecule. Again, we have these amine groups here that are connected to iron, okay? So it's interesting. It's just an interesting, I mean, these are both functioning parts of a biological system. One is animal and one is plant, but they're both, they both involve amino acids. So all living cells produce amino acids. In the case of animals, they cannot produce all types and they have to be provided in the form of a diet. Guys, I can't hit the button the right way. I just struggle. Down is forward. Okay, so this is an example of amino groups, amine groups, or specific amino acids that and their function in plants. Proline, hydroxyproline, stressors, cysteine, glutamine, glycine, histidine, lysine. These names probably are familiar to you, but specific amino acids are built by the plant for specific functions and specific needs. So I really like this slide because it talks about why an amino acid fertilizer would be of value. So the nitrogen cycle is incredibly taxing, energy taxing on a plant. So there's a lot of reduction. Vic was talking about, some reduction chemistry, nitrates, ammoniacal nitrogens, ammonium forms. To convert those, reduce those, is very taxing on the plant system, energy-wise. There are some other nutrients involved in nitrogen reduction. You need mag, you need sulfur, copper, iron, zinc, and moly. Manganese is part of that. That little battery symbol down there is a symbol for phosphorus. And of course, our most important macronutrient, water. Reducing nitrogen costs plants a lot of energy. So what if you could minimize that? So picture a plant that wants to reduce a raw material or build from a raw material and create something. Because what plants want to do is they want to use nitrogen to build amine groups and begin the, thousands of plant-built products starting with nitrogen. So a plant wants to build a bicycle, okay? We're going to say the end product that that plant wants to build is a bicycle. Maybe that's an enzyme. Maybe that's a more complex, peptide, short chain, something that involves amine groups. If you begin the process, so into the factory comes round steel tube and wheels, okay? So now the plant has to bend the pipe, cut the pipe, build the frame, mount the wheels, find a bicycle, find the seat, find the handlebars, and it takes energy to do all these things. You picture an assembly line of workers working on this bicycle. It costs the plant energy. But what if walking in the back door of the bicycle factory is a pre-built frame, not just round tube, and maybe it's got wheels on it. Now that plant can take that pre-built bicycle frame and start adding, okay? This is the reason why pound for pound, and there's been a lot of work done on this, pound for pound, the same nitrogen test. Remember, when you see a nitrogen number on an amino acid, it's not really a nitrogen number. That nitrogen is complexed as an amine group of some type. All those different types of amines or different types of amino acids are present in an amino acid profile. So that amino acid comes in, it wants to build a bicycle, it comes into the factory as a frame with wheels on it. It takes so much less energy to get to the bicycle if it comes in pre-built. Pound for pound, the same test nitrogen number on an amino acid, if it's plant available, 10-to-1 efficiency. Now, mind you, these products aren't easy to, they're not inexpensive like urea fertilizer, but if you think about them, and I'm not saying urea fertilizer is inexpensive, but relatively, these things are more costly to purchase and apply, but they come in 10x more efficient. This is why they're cool. I'll figure this thing out one day. So this is an example of an amino acid profile from a soy hydrolysate. So we look at these things and this lab actually quantifies all those different amino acids that are present in a soy hydrolysate. Again, they all have specific, plants can build specific things from them because they're all just a little bit different. But that just is an example of what a amino acid profile looks like. This is a really cool little bit of work. Now, I don't know who did this work, but I found it a couple years ago. And this is where I mention that these types of nutrition are very plant friendly and accepted in a plant tissue system. So what they did was they've got an untreated control here. This is what I like about this. This is pepper plants, and it was a 2-year study. So what they did with treatment 0 is an untreated control. With the first treatment, they used a 2% micronutrient solution. So they used soluble micronutrients. I don't know what the array was, and it was 40 ppm of the micronutrient. on its own. That's T1. T2 is only an amino acid application. T3 is a 1% amino treatment, 20 ppm of each micronutrient. T4 was 1.5% amino. So this is probably just a nitrogen amino. So then they ramped it up to 2% with treatment 5. So look how these plants responded to adding the aminos. So here's an UTC, you're not going to see much there. This is the micronutrient only, and I'm assuming this is in a somewhat sterile medium, soil medium. So now you start adding, this is the amino by itself, but look what happens, and this is yield. Look what starts happening to this yield number 2 years in a row when you combine the amino acid with a micronutrient application. Aminos drive things into the plant and make plants more efficient. And they utilize, and this is just an example of that. So I've gotten tons of experience doing foliar sprays with these guys. That's mostly what we've focused on because they fit really easy. They're 1.5 pounds per acre, kind of 2 pounds. These low use rates, 4, 6 pounds, whatever. This is some work we did in hazelnuts. And we had some real problems here in this field. Anyone here familiar with sap testing? Good for you. So you share my pain and struggle with the amount of labor it takes to do sap testing. Blueberries are a joy because their leaves are about that big. And it takes a lot of them to make a 100-gram sample. Hazelnuts, not so bad. They're nice big fat leaves. So this is a single treatment of a product called Amino. So that's a calcium paired with the amino acid. This is one treatment. I think it was about 6 pounds. 1 month apart. These samples are taken 1 month apart, so before and after. And it's incredible how far you can push some of these in the plant tissue. This is in the leaf, mind you, not necessarily in fruit or whatever. But that's what we found. This test is MAG. Again, struggling with MAG, but moved it really quickly up into, a minimum area. It was truly deficient. But what's fun about sap testing is that it is so quick, and you can do an application and see within a week things shift. And this is just proving that same slide, that pepper slide, that you can move with aminos, you can move micronutrients really quickly. Here's a wine grape trial. Again, changed the yield dramatically. And this, these aren't really big use rates. And there's, more information here. This was, I think, a Pinot Noir grape. And really quickly, you can move numbers up. This is a measurement of YAN. So that's called, for all of you wine grape people out there, it's called yeast assimilable nitrogen. And it's a big part of fermentation. And, what is in that grape that's going to turn it into wine? They don't like to add things like sugars to wine production. They will in order to get things to ferment correctly. But if you have a high YAN number, you're going to make better wine and you're going to make it cheaper. So that was just a fun trial. Yeah, and this is just about our aminos, soy protein hydrolysates, non-GMO, made from non-GMO soybeans and enzymes. Vic could tell you more about that. And yeah, all the stuff's guaranteed. Anyways, that's a rough description of amino acid fertilizers. Does anyone have any question? I think that's the last slide. Yeah. Anybody? I kind of want to ask a question, and I've met you, but I can't remember your first name. The guy behind you, sorry. Barry? Can you tell me your story about using amino fertilizers? Or a quick? I'm talking to the gentleman right behind you. Yeah. Andrew. Don't ever point at people in the crowd. They think. There you go.
All right. Okay. Yeah, Andrew Mans. So we we grow in an acre of greenhouse as well. So
Right.
and we do veggies, so higher value crops. So and I am certified organic. So of course the aminos have been part of my crop. Inputs on the greenhouse are about 600 gallons a year on 1 acre of greenhouse, but it's high yielding tomatoes, peppers, cucumbers. So it becomes a very like fish. And we use some of the soy ones more for foliars, like you mentioned, but becomes a very stable weekly input, and it drives a lot of the microbial activity and also a way to go in with other nutrients. So works quite well.
Good. Thanks for that, Andrew. Now I'm remembering meeting you last year, that learning that you were growing in biological soil in a greenhouse. Yeah, that's great. Any other questions about amino acids? Okay.
One question for the amino acids, there's such a broad spectrum of them, and then you're talking about fertilizers. How do you distinguish which is right and which environment do you use? You guys have a technology of adding minerals to the amino acids. How do you know that, like, what is right? Like, it's a big, what is there, 16 essential, and then you have if you're adding minerals to the aminos, how do you know where you're at in all of this?
Well, basically how we know where we're at, and Vic, wherever you are, you can jump in. That amino acid profile that I showed, we don't pick and choose that. That just is the amino acid profile of the non-GMO soybean that our product is built from. So head toward the micronutrient added products, it's just as a percent by weight. So the specific numbers, I know CalAmino's an 8%.
Yeah, I think. Can you guys hear me? Without microphone. So amino acids are good complexers. They're not really strong chelators, but they're complexers. So, for example, EDTA is ethylene ditetraamine, the two groups of amine that comes together, right? And they pretty much make a bond that cannot be broken. Like you have zinc EDTA that's connected with two amino groups, right? And then as soon as zinc goes away, calcium replaces. That's why there's bone loss in the body. That's why they don't allow EDTA as a human supplements, right? They shouldn't apply it in the ag either. But aminos are softer complexing agents. So they have limited amount that you can complex them with. I think our top line product in that category is a 8% calcium. And beyond that, it won't fit in that complex. So 8%. Sorry, 800 10% calcium, but you can't put more calcium in there. However, if you start pushing zinc in it in the matrix, you can go much higher zinc, and you can say like almost 100 15% zinc, because it'll blend better with the amino as a complex. So the products we have currently in the US are 800 10% calcium, 500 5% boron, 200 14% potassium, and then a multiple amino acid complex. And they're all organic. But they all go in high value crop, right? I think the low input crop system, the most unique product we have is calcium amino acid complex. Does that answer your question? Where did Bill go? Oh, you're.
Any more questions?
Otherwise, Lauren. Well, we are available afterwards, guys.